Hello, Biochemistry 2 students. This is the weeks 3 and 4 video. I'll jump right in. So during week 3 of the course, we spent a good bit of time looking at different amino acid side chains and the amino acid sort of backbone section, which is on the right side of this slide in the red box. We talked about how an amino acid is really an amine group with a carboxylic acid group, both linked to a central carbon, which has both a hydrogen linked to it and then a variable side chain, which would go right in this R position. These are sometimes referred to as R groups. These side chains have different properties depending on their atomic composition. Some of them, especially if they're pretty much all made of carbon and hydrogen, like alanine or valine or phenylalanine, or if they're, mostly like pretty overwhelmingly hydrocarbon, like tyrosine, for example, then those are nonpolar or hydrophobic or fat soluble. Call these nonpolar or fat soluble side chains. Okay. And that gives those amino acids particular properties that we'll come back to in a minute. Or if there's quite a bit of nitrogen and oxygen in the side chain, sorry, circling too much there. If there's quite a bit of nitrogen and oxygen in the side chain, or, compared to the amount of hydrocarbon, there's, enough polarity present, such as an OH bond or an NH bond or a CO bond, then you have polar or water soluble, or hydrophilic, if you prefer, hydrophilic, water loving side chains. If you have positive charges, as in these, or a spot where a positive charge can go, such as here, then you've got positively charged side chains, which can also be called ionic side chains. You could also have potentially negatively charged side chains, also referred to as ionic side chains. There are three that are kind of like, sort of special cases. Glycine, because it's just a hydrogen, is very, very small. Generally, it's considered hydrophobic, but it's just really little, so it can kind of get away with going a lot of different places. Proline is a really funky one because the side chain actually goes back and sticks to the amine group, so it kind of loops around. In red there is the side chain. So that's sort of a funky and weird one. And then cysteine, which can kind of behave multiple different ways, is another funky and weird one. Those are not ones I'd ask you to really make a call on. The rest of them, I think, are pretty straightforward in terms of determining, are they polar, non-polar, or are they positively or negatively charged? Now, the reason why any of this matters is, well, first of all, it's an application of electronegativity, which is a property that we emphasized in the periodic table. And it's an application of electronegativity that then allows us to think about how amino acids form proteins. So in order to form a protein, amino acids have the acid group, the carboxylic acid group, which is here circled in red, from one amino acid, linked to the amine group of the next amino acid in a row by dehydration synthesis, the removal of water, which should be a familiar reaction to you from last year. When dehydration synthesis occurs, it creates a bond between amino acids that is called a peptide bond. And if you have multiple peptide bonds, you've got lots of amino acids linked together in a row. That's called a polypeptide, which is another way of saying protein, essentially. Now, each of these amino acids, of course, has its own side chain. In this first image here, and really these should be R's as well. I don't know why these are being put there as H's. This should be consistent and call them R groups. They're variable sections. So here they're just being shown without any detail as like, yeah, that's a side chain. We're not saying which side chain it is. It could be any of the ones from the previous slide, right? It could be any of these. In the image on the right side of this slide, we are being shown the actual side chains. This is CH3, which is the alanine side chain. Alanine is one of the 20 amino acids. This is a hydrogen, which is glycine. Glycine is a different one of the 20 amino acids, right? And so this would be the beginning of a protein whose primary structure being the order of amino acids, is alanine followed by glycine. Okay. So as you link together amino acids into a primary structure, a chain of amino acids, here they're each shown as like a colored little circle, that primary structure then folds up. It folds up. We did a modeling activity using bendable tubers and little plastic side chains in class. As it folds up, one of the things that happens is that all of the hydrophobic, all of the nonpolar, all of the fat-soluble side chains, those all end up like going kind of toward the center of the protein. We would call that center the hydrophobic core of the protein, right? And there they would be buried away from water. Now, if you imagine like maybe the actual locations of those, like maybe these four that I circled in red are nonpolar, and then maybe this one is nonpolar, and maybe these two are nonpolar, and maybe this one is nonpolar. And so you'd have to imagine that when this protein folds, it's going to have to be manipulated into a manner that puts those chunks together in the middle. And that might be why it folds into some kind of contorted shape. And that shape may look like nonsense. It may look like a pile of spaghetti, but it's actually based on very specific rules, right? And so it's a very consistent shape. Whenever you have fat-soluble amino acids in these positions, it's going to fold in this way predictably in order to get them to the center. And so it's going to make a consistent shape, even if that shape looks kind of crazy. Similarly, the backbone of the protein, that's the part with the amine and the carboxylic acid, which is all throughout the chain as well. That part can actually form its own kind of little interactions through hydrogen bonding in the backbone. And we refer to that as secondary structure. There are two different main types of secondary structure. There are spirals called alpha helices, or as a singular is alpha helix. Alpha helices is the plural. Or these kind of zigzaggy sort of sheets, which are called beta sheets, which are also stabilized by hydrogen bonding between parallel parts of the backbone. Okay, so those are secondary structures. Those two structures together, the sheets, the helices, and the R group interactions, which is the tertiary structure, create the overall structure of that folded up chain, that folded up primary structure. And then potentially, in some cases, that folded up structure can interact with other chains that have been folded and make what's called a quaternary structure, where multiple different polypeptide chains adopt secondary and tertiary structure and then interact with each other in what's referred to as quaternary structure. What's amazing about this, right, is that all of the proteins that make up organisms, and we learned about them last year, they're coded for by genes. And what the genes are coding for is a sequence of amino acids. So if you look and you see, oh, phenylalanine, followed by valine, followed by asparagine, followed by glutamine, and so forth, really there was a gene that said 3 letters at a time, phenylalanine, then valine, then asparagine, then glutamine, and so forth. Okay, so that is what the gene is coding for. The gene codes for a primary structure. And then based on the primary structure, the protein folds into secondary, tertiary, and in some cases, as in here, where we've got 2 different chains, one in blue, one in red, sticking together, that's quaternary structure. Now, all of that folding makes a shape that, again, may look kind of crazy, but is very consistent. In this case, the shape that you're looking at is insulin. So insulin, you all have genes that code for insulin. And as your genes are transcribed in your cell's nucleus, and then translated on ribosomes into protein sequences, what's actually happening is the genetic code is coding for the order of amino acids, which fold consistently into this three-dimensional shape, which makes you able to have the functionality of insulin, because it is the shape of it that actually gives it its function. Structure determines function. And to really get you to see this, here's another way of looking at this. In the top right of this slide, in kind of a tan, sort of yellowish, almost orangey color, is insulin. That's a different three-dimensional sort of space-filling model of it. And this is a picture of insulin based on its shape, fitting perfectly into a groove on the side of what's called the insulin receptor, which is shown here in blue. That's a protein that you also have genes that can code for, and that then that protein is embedded in the cellular membrane, where you see all these phospholipids, which you'll remember from last year, with their hydrophobic tails, fat-soluble tails, and their hydrophilic head sections, right? So this is how insulin sticks to its insulin receptor, which is embedded in the cell membrane, and causes it, by sticking there, to change shape, snapping these two sections together. And when they snap together, they initiate a response inside of the cell. So literally your ability to build the shape of insulin, which is genetic code, is what allows you to then have the function of that shape sticking to its receptor on your cells, changing the receptor's shape, and, as I go to my next slide, initiating a series of changes inside of the cell. Insulin sticks to its receptor. The receptor changes shape. Here there is less detail being shown, but you've seen it on the previous slide. That initiates a chain of events that result in glucose's channel protein being shipped to the cell membrane. So what is insulin's job in your body? It's to tell your cells to take glucose out of your bloodstream and pull it into the cell. Glucose can't go across a cell membrane because glucose is polar. You see how all the things we learn, like they have meaning, and if you can follow them, you can really understand a big story here. Glucose is polar, so it cannot go across the membrane. It is repelled by the phospholipid tails because they are fat soluble. Its only way in is through this channel protein. And what insulin does, by its shape, it sticks to a receptor, which interacts with another shape, 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 which pushes glucose channels onto the surface of the cell, where they allow glucose to flow into the cell. And so this is how insulin is making your body right now. If you've eaten anything with glucose in it recently, that would include anything starchy, anything with sugars in it, then your body is releasing insulin into the bloodstream. It is sticking to cells at the insulin receptor based on structure. It is initiating the function of pushing glucose channels to the surface of those cells, and that is facilitating the movement of glucose into those cells, lowering your blood sugar, bringing it back into a safe zone, getting the glucose from your food out of your bloodstream and into your cells where it can be useful. That's just one function. Now imagine all the genes you have and all the functions that their structures can catalyze, and you're understanding a biochemical system. The great thing is it's not terribly complicated. It's basically the same elements, FONC, over and over again, arranged into the same molecules, carbohydrates like glucose, lipids like phospholipids, proteins like insulin. Genetics make sure that the right shapes are built, especially shapes that are protein-based. And those shapes then execute these functions of facilitating certain movements, certain changes, certain interactions between the molecules in the system. Okay. You can almost think of this like an ecosystem. You know, when you have more wolves, you have fewer deer. When you have more insulin, you have more glucose channel proteins in the cell membrane, right? It's a similar sort of regulation, except that instead of one species regulating another, it's one protein regulating another. Still, a cellular system is just like an ecosystem. It's just a very small ecosystem where the interacting entities are not species, they are proteins. All right. So I think that's a pretty good kind of crash through this. You might need to watch this video a couple times if you didn't totally follow that, but I think it's a really good way for you to start thinking on a high level about biochemistry content here in your second year of thinking about it. Hopefully really understanding how a biochemical system functions, what the basis of that functioning is. Thanks, everybody.